Hi there and welcome to this new channel about the amazing world of neuroscience. Today we will talk about one of the most prevalent and crippling diseases in the West, namely depression or major depressive disorder. It is so prevalent that at this moment 1 in 6 people in the US are currently suffering from it. Even more astonishingly, the chance of recovery is actually very low for depression, being 50% during the first episode and as low as 20% during or after the second episode. But have you ever wondered why it is so difficult to just stop being depressed? Well, I know I did when I was suffering from depression. The answer to why depression seems so hardwired is because basically it is. Namely, the brain changes chemically and physically the longer and more severe your depression is. While you may have heard that depression is associated with a chemical imbalance of neurotransmitters such as serotonin, norepinephrine and dopamine in various brain regions, there are more and more studies that show that depression is also related to various changes in brain structure. This makes it all the more clear that it may become increasingly more difficult to work through depression, such as many people who suffer from it for a longer period of time may experience. The manner in which the brain structure changes in depression depends on various variables, and the changes are not the same for all individuals suffering from MDD. It is thought that the most influential variables are the severity of the illness, the type of symptoms, and the duration of the illness. Other factors could be demographics such as age, the genes, and comorbidities. The brain structure may change in manners such as neurons reducing their size, neurons dying or not regenerating, the connections between the neurons or synapses are being weakened or strengthened, or the ability of the brain to regenerate its functions decreases. These changes can happen in MDD because of the excessive amount of stress hormones that are associated with this mental illness. Excessive stress hormones are known to cause inflammation, lack of oxygens or nutrients, oxidative stress, and excitotoxicity, which can all damage brain tissue and neural connections. It is also known that these events can be directly responsible for various symptoms of depression. So what causes this excess of stress hormones? Let's look at one of the most important brain areas for the management of stress, the amygdala. The amygdala is part of the limbic system of the brain, which consists of multiple interconnected areas. There is no scientific consensus about which areas belong to the limbic system, but the more commonly referred areas are the amygdala, the hippocampus, some parts of the thalamus, basal ganglia, and the cingulate gyrus. The primary role of the limbic system is to deal with behavioral and emotional responses and processing, and it is also involved in the formation of memories and learning. The amygdala has a key role in all of these functions, but one of the most outstanding roles of the amygdala is the assessment of potential threat. The amygdala is ultimately the part of the brain that decides whether something should be considered dangerous, and through these connections with other brain areas, it could elicit the fight, flight or freeze response. This response is known for elevating the heart rate, the alertness and stress hormones in the body, which ultimately cause fear. There are 2 neural pathways that your brain can use to check whether something is a threat or not. There's the short neural pathway that only collects the basic sensory data about the threat and sends these sensations to the amygdala. There's also a longer pathway that goes through multiple more complex areas of the brain, such as higher sensory areas or areas that may recollect memories or have an executive function. This pathway is more accurate in threat prediction due to the amount of context it can process. In some cases, it is more valuable to take the quick route to directly assess the threat. This is the case, for example, when you encounter a toxic snake. Your amygdala would make you use your fight or flight response directly through the short route, avoiding anything that may potentially lead to your death. In this case, it is not so devastating if your response is not so accurate and that the snake turns out to simply just be a rope. It would be more devastating if you would not react quickly enough. However, in most modern situations, the short route of threat detection is not very useful. Modern threats are way more complex than the threats that exist in nature. Think for example of a math test, public speaking, or encountering someone or something related to traumatic past experiences. Labeling these events as threats will cause an increase in stress hormone production, as well as a change in neurotransmitters. How much your stress hormones increase depends on the severity of the threat and also on your own brain, as it has been reported that stress responses are significantly larger in patients with MDD. And in approximately 40 to 60% of depressed patients, the cortisol level stays high for much longer than in healthy individuals. In terms of structural change, patients with MDD show a widespread reduction of the connectivity between the amygdala and many brain regions involved with emotional processing and emotion regulation, including the ventrolateral and the dorsolateral prefrontal cortex, the occipital lobe, the cerebellum, middle and superior temporal regions, as well as deeper structures as the insula and the caudate. These connections are crucial to form proper and emotional responses and to correctly identify possible threats through the long and the short route. This decrease of connection is probably one of the reasons that the emotional responses of patients with MDD are not always appropriate. A decrease of functional connection between the amygdala and the ventrolateral and dorsolateral prefrontal cortex are also related to relapse. On the flip side, many studies report that the left amygdala may become hyperactive in patients with MDD and PTSD compared to controls. This increased activity is thought to be related to heightened emotional responses, particularly to negative stimuli. A study from 2022 suggests that heightened amygdala reactivity to fearful and angry faces is associated with the future emergence of self-reported mood and anxiety symptoms in young adults. There are also studies done in patients with PTSD that showed similar results with faces and explosion sounds. Finally, there are also multiple studies that suggest that the amygdala also shrinks in size in patients suffering from depression, especially in patients with recurrent MDD episodes and suicidal or psychotic symptoms. Other studies suggest that the amygdala can also increase in size, especially in those patients using antidepressant medication or with the disease onset in childhood. Another area that could significantly change its structure is the hippocampus. The hippocampus is also part of the limbic system of the brain and is located near the amygdala. Its primary roles are the encoding and consolidation of memories, spatial navigation, and learning. It is also known to be essential in emotion regulation and in forming appropriate emotional responses. The hippocampus also stores context-specific information of threat memory, which is relevant for the long route of threat detection. Interestingly, a change of hippocampal structure is considered to be one of the most prominent biomarkers of MDD and anxiety disorders. The hippocampus is consistently found to be significantly smaller in patients with MDD compared to healthy controls. A study from 2020 argued that the decrease in hippocampal volume is proportional to disease duration, and another study established that the volume is only significantly decreased in patients with more than one depressive episode. While a smaller hippocampus is often found in patients with MDD, hippocampus volume also seems to be predictive of MDD onset and remission. Importantly, the hippocampus is only one of the 3 areas of the brain where new neurons continue to develop from the stem cells in the mature brain. However, stress is known to impair the growth of new neurons in the hippocampus. The reduction of hippocampal cell growth is related to a decrease of learning ability. The next area that we are going to cover is the medial prefrontal cortex. The medial prefrontal cortex is a so-called higher order area, as it is involved in complex cognitive processes such as decision-making, inhibitory control, executive control, conflict monitoring, and memory. It has also a large role in emotion regulation, self-reflection, regulation of negative affect, and sociability. Interestingly, the medial prefrontal cortex is strongly connected to the limbic system, and it plays an executive and inhibitory role in threat detection and behavioral responses. The medial prefrontal cortex also plays an important part in fear conditioning and fear extinction. Unfortunately, it is also commonly reported that this area can be affected by stress and MDD. Like the hippocampus, it has been reported that the medial prefrontal cortex volume can be reduced in patients with MDD depending on the duration of the disease. It has been hypothesized that the reduction of medial prefrontal cortex volume may be due to the significant reduction in the number of glial cells in the medial prefrontal cortex in MDD patients, which provide energy for the neurons. It has also been found that the inflammation and oxidative stress levels in the medial prefrontal cortex are much higher in patients with MDD than in control groups. The connectivity between the medial prefrontal cortex and other brain regions is also commonly altered in MDD. One study showed that the severity of MDD is related to the connectivity of the ventral medial prefrontal cortex with all other areas of the brain. Namely, a higher severity would mean that the ventral medial prefrontal cortex would be less connected to other brain areas. The last one in today's list is the anterior cingulate cortex. It is responsible for a number of cognitive functions, including emotional expression, mood regulation, and attention. It is also known to severely reduce in size and density in patients with MDD, primarily seen as a decrease in gray matter volume. A decrease of glial cells in the ACC is also prominent in MDD. Early life trauma and cumulative adversity, or adverse social and non-social environments, are also correlated to decrease in ACC size. ACC dysfunction has been strongly implicated in depression in both adults and adolescents. During emotional processing, self-regulation and response to stress, patients with MDD seem to have more abnormal patterns of ACC activity than controls. So today we talked about 4 brain areas that can be structurally affected by MDD. This is by no means an exhaustive list. And unfortunately, there are many more physical and chemical disruptions in the brains and bodies of patients with MDD. It is important to remember that it's a very complex disease and the disruptions may differ per person and per trauma type. One critical question arises, however, namely, what can we do to stop or revert these structural changes in the brain? The answer to this is complex, as it depends on the type of damage and the clinical case. Additionally, not a lot is known about this line of research. However, we do know that at least some structural changes can be reversed by treatment of the clinical symptoms and by lowering stress hormone levels. This is the only way to stop the neural damage that is caused by stress, and this will cause your neural pathways to be altered to a more normal state. The most efficient way of overcoming the patterns of MDD is working through it actively with a licensed professional who can find the reason behind your depression and tackle it accordingly. As each person's depression is rooted in a different problem, there is no easy approach to this. However, there are 3 types of treatment that can be used in MDD, and the combination is often more effective in more severe cases. There's bottom-up, top-down, and medication. The method that is most commonly used with the absence of strong trauma is top-down therapy, which focuses on making the mind more aware of its behavior and thoughts. This is often achieved by talking to the therapist and writing down triggers and thoughts. One common method is cognitive behavioral therapy. The opposite approach, namely the bottom-up approach, focuses on the integration of sensory information. The flow of sensory information can get stuck in the same neural pathways that may lead to stress and fear, and the bottom-up approach may change these pathways. This method is often recommended if physiological issues may occur due to the trauma, such as asthma, difficulty breathing, panic attacks, sleep problems, fatigue, muscle aches, headaches, and more. Examples of this approach are EMDR therapy and sensory-motor psychotherapy. Medication can also be used in some more severe cases, of course only if your psychiatrist agrees upon its use. While there are multiple reports that antidepressant medication can stop or reverse neural decline and increase the development of new neurons in the hippocampus and the prefrontal cortex, medications should always be used with caution, as there may be a number of side effects. One method that can be considered to be both top-down and bottom-up is mindfulness meditation. It has been suggested that mindfulness results in top-down emotion regulation in short-term practitioners, and bottom-up emotion regulation in long-term practitioners. Some of the main neurocognitive mechanisms implicated in mindfulness meditation include attention control, emotion regulation, and self-awareness. Because of the relatively quick results that one can get from mindfulness and the positive effects that it has on mood, it has become increasingly more popular in the medical field. So I hope you found this video to be informative. If you liked it, hit the like button and subscribe if you want to learn more about various neuroscientific topics. Have a great day. Bye bye.